unit totally have completed in the that unit is totally complete in the last class. Okay. Okay. Now we will share the first. Today we will discuss about to the nitrogen metabolism. Nitrogen metabolism. What is the nitrogen metabolism? How to use the plant in the atmospheric nitrogen? How much of nitrogen are there? These are things we will discuss today's class. Okay. Now I'll share my PPT. See, nitrogen metabolism. What is nitrogen metabolism? What is the use for nitrogen metabolism in plants? This is the what type of micro element or macro element? How to absorption of the nitrogen in plants? Where you can availability are there? These are things we will discuss today section. Okay. Now, today start to the nitrogen metabolism. What is the nitrogen metabolism? All living organisms are basically composed carbon, hydrogen, oxygen, nitrogen, and many other chemical elements. Okay, these are elements are the most important. All living organisms in the plants, animals, anywhere living organism are basically composed of carbon, hydrogen, oxygen, nitrogen, and many other forms of the chemical elements. These elements contribute to finally organize various biomolecule present in a cell. Present in a cell. Nitrogen is next carbon in importance in living organisms. Okay. In a living organism, living cells, nitrogen is an important constituent of amino acid proteins. You can observe the nitrogen, where you can observe that one. That is the important constituent of amino acids, proteins, enzymes, enzymes, vitamins, vitamins, alkaloids, and some growth hormones. You can observe the nitrogen is the like this. Okay. Now, therefore, the study of the study of nitrogen metabolism is absolutely essential because the entire life process become the entire life process independent on the nitrogen containing dependent on the nitrogen containing molecules. In this lesson, you will learn about various aspects of nitrogen metabolism, include nitrogen fixation and nitrogen assimilation in plants. Both these are things you have studied in this topic. Okay, in the nitrogen metabolism time, you can study it to the in this total process. And here now we will start to the what is the molecular nitrogen. Okay, here in the atmospheric nitrogen, only 70% nitrogen is there. How much? Only 70% nitrogen is present in the atmospheric nitrogen. Nitrogen is the primarily present in the atmosphere freely as what position is there in nitrogen? That is a dinitrogen or nitrogen gas. How to you can observe this is a dinitrogen or nitrogen gases. It is present in the combined form as sulfate or sodium nitrate, sulfate or sodium nitrate. And uh, child in South America, the main South America is the major source of this nitrate and nitrogen. Molecular nitrogen or diatomic nitrogen. Molecular or diatomic nitrogen N2 is a highly stabled. Which one is the stabled is there? N2. N2 is the stabled nitrogen. N2 is the stable bonded molecule nitrogen. These are diatom. This is the because of the N triple bond N is the mostly triple bonded structure is there. In that triple bond is the stable nitrogen. The molecular nitrogen as such is not very reactive in the atmosphere under normal conditions. Under normal condition. In the atmospheric molecular nitrogen is in the atmosphere 75, minimum 78 to 78.3% by volume in a very low boiling point. What is the boiling point here? You can observe minus 195.8 degrees centigrade, which is evenly lower than that the oxygen. Proteins present in living organism contain about to the 16% of nitrogen is there. Proteins is the present, 16% of nitrogen is present. What is the nitrogen cycle? Okay, in the nitrogen cycle, here in the nitrogen, main is the source of nitrogen is the main important in the atmosphere found in the like gaseous form and measure about 78.8% in value. This elements nitrogen is not available in higher plants, but certain prokaryotes. What is a certain prokaryotes? That is blue-green algae and bacteria can utilize it. However, higher plants can utilize nitrogen only. Nitrogen only in combined form. There are following four nitrogen sources are there. The first source is called the nitrate nitrogen. Nitrate nitrogen. Second one, ammonia nitrogen. Third one, organic nitrogen. Fourth one is molecular nitrogen. These are the four sources. First one, nitrate nitrogen. Nitrate nitrogen, nitrogen in soil. Nitrogen in soil is found mostly in oxidation from nitrate. Nitrate NO3 minus is observed by the roots of higher plants and is transported to young leaves where it gets reduced to ammonia, that is NH3. The reduced ammonia reduce taken place in two steps. The first step, reduced to the first step takes place in cytosol, where nitrate is reduced to nitrite by the enzyme called nitrate reductase enzyme. The second step takes place in chloroplast, where nitrite is the NO2, nitrate is the NO3. See, listen careful. Nitrite is the NO2, nitrate is the NO3. Reduced to ammonia, NH3. The enzyme is called nitrate reductase enzyme. Next one, ammonia nitrate. The molecule, the ammonia nitrate. Ammonia nitrogen is in the soil, is also found in the reduced from ammonia. NH4. NH4 is observed by the root of higher plants and participate higher plants participate directly in metabolism. The source of ammonia in the soil is artificial introduction. Is the artificial introduction as a chemical fertilization or microbial action upon dead and decaying organisms. Paddy plants are known to absorb the ammonia rapidly. NH4 is rapidly. Next one is the organic nitrogen. The soil may be also organic nitrogenous compounds such as urea, amino acid, etc., which are formed due to the decomposition of dead and decaying organism by activity of microbes or when farmer add chemical chemical fertilization to the field. The roots of higher plants take up meaning absorption of nitrogen. These organism nitrogenous compound which participate directly in plant metabolism. This is the third one, organic nitrogen. And fourth one, molecular nitrogen. Molecular nitrogen is absorbed by the only prokaryotes, such as N2 fixation bacteria. N2 fixation bacteria, Azotobacter, Rhizobium, and blue-green algae, Anabina, and Nostoc. Higher plants cannot use molecular nitrogen present in atmosphere directly. Prokaryotes observe nitrogen and reduce it to NH3, which in turn is catalyzed by the nitrogenase enzyme. This is the four, nitrogenous molecule. Now we will discuss about to the nitrogen cycle. What is the nitrogen cycle? How to use the nitrogen cycle? These are things we will discuss. Okay. Nitrogen cycle. Nitrogen is essential for life. In the cycle, nitrogen is essential for life. Essential for life. Essential for life. As it is the part of nucleic acid, amino acids, proteins, enzymes, vitamins, and chlorophyll. No doubt that nitrogen makes up the greatest part of the atmosphere. But green plants can use little or no atmospheric nitrogen N2 in their synthesis. Green plants can use nitrogen as ammonia. See, you can observe this one. Okay, green plants can use nitrogen ammonia. They can use little at no atmosphere nitrogen in their synthesis. Green plants can use N2 as ammonia, N3, or in the form of nitrates, salts containing NO3, and these are obtained from the nitrogen fixing bacteria of soil, which can utilize the N2 directly in the synthesis of their own amino acid and proteins. Some nitrogen fixing bacteria live in the root nodules of family, Leguminosae family. That is called symbiotic bacteria. Example, Rhizobium, Melotti, Rhizophyrium trifoli, etc. And some bacteria, other microorganisms live independently in the soil. That is called non-symbiotic independently living bacteria. Example, Azotobacter nitrosomonas. With the Byseringia, certain species of fungi and blue-green algae. In either condition, location, their death-free nitrogen compounds which can be utilized by other plants and especially green plants. The soil bacteria meaning nitrosomonas bacteria. Nitrosomonas is the soil bacteria. Oxidized ammonia to nitrates. Other soil bacteria, nitrobacter, etc. Oxidized nitrates to nitrates to nitrates, nitrite to nitrate, nitrification. Further nitrates and nitrites are also produced from atmospheric oxygen and nitrogen by lighting flashes. You can observe when the rain, before rain, lighting flashes is there. In that time also to fix the nitrogen. Rain then carries such nitrates to the ground. Some of the environmental source of nitrate are well known, such as mineral accumulations through dissolution of rock by the water or through addition of soil fertilizer by man. The N2, nitrogen of green plants, thus acquired from ammonia and nitrate in the soil from the nitrogen fixing bacteria may be passed on to animals, may be passed on to animals also, passed on to animals, passed on the animals, green plants, either condition, location of the death of free nitrogen compounds. So next one is the further nitrate production, atmospheric oxygen. Some of environmental source of nitrates, some of the environmental source nitrates are well known, such as mineral accumulation through dissolution of rock by water, through addition of soil fertilizer by man. The nitrogen of green plants, nitrogen of green plants, thus acquired from ammonia and ammonia nitrates in the soil or plant soil or form nitrogen fixing bacteria may be passed on animals that eat plant, and then from animal to animal, meat eating animals. That is the animal to animal meaning. Meat eating animals is the that one. Meat eating animals. Nitrogen is extracted by animals in the form of urea and also is found in the dead bodies of organism. Again, this is only the function of bacteria which return this locked N2 to the cycle. Bacteria of decay produced ammonia from proteins and other nitrogenous compounds. Other bacteria, nitrifying bacteria, oxidized ammonia to nitrates, and still other bacteria compound to bacteria do further oxidation to nitrates. In such a way, inorganic nitrogen compound directly consumable to green plants are restored in the soil. On the other hand, facilitations. On the other hand, facultative and facultative, facultative and anaerobic bacteria of the soil. Facultative and anaerobic bacteria soil, denitrificating bacteria such as pseudomonas, break down nitrates and some other compounds and liberate nitrogen. After nitrogen, N2 is at last incorporated in plants as protoplasm, etc. It is converted into animal tissue. Animal tissue meaning animal proteins. When plants and animals die and their wastes decay, the saprophytic microorganism change back into the form of ammonia and other nitrogenous compounds, and thus the cyclic recommences. The N2 cycle in brief can be assembled by the made up of the four important characters are there. See, this one is the nitrogen compound. One is the transformation atmospheric, and next one biological. It is the N2 cycle. This is the N2. This total is the N2 cycle. This N2 cycle, four important processes are there. The first important process, the transformation of atmospheric gaseous nitrogen. What is the first one? This is the atmospheric nitrogen, transformation of atmospheric gaseous nitrogen by microbes and certain photosynthetic bacteria and blue-green algae into the combined from NH4, nursing bacteria and the green plant. This process is generally called biological nitrogen fixation. Next one, the transformation in soil of organic nitrogenous organic nitrogen compound, that is a arising from decompositions and autolysis of all forms of biological substance, or from extreme excrete of animals, or forms products of metabolism of living soil organism into NH4 ions. This process happen to take the various microbes which may be aerobic or aerobic and anaerobic. They break down the organic nitrogen compound into NH4 ions. As a part of their metabolism, formation NH4 ion may be due to the action of hydrolytic and oxidization enzymes found in the microbes. This is the second process. The third one, the conversion of ammonium, the cations to nitrate and nitrations. This process is called soil nitrification, which is completely two types. Soil is the completely two types. One is the nitrosomonas, converted NH4 ions into nitrates, and nitrobacter converted nitrates into nitrate. The reduction of nitrates into faculty finally into NH4, NH, NH4 ions are into free molecular nitrogen, which goes back the atmosphere. This is caused by the various microbes. These are the N2. This is the N2 cycle. In brief, can be summarized the four steps. Now we will discuss about the biological nitrogen fixation. Biological and nitrogen fixation. Biological nitrogen fixation, here two are there. One is the abiological, second one is the biological. What is the difference in between abiological and biological nitrogen fixation? The first one, the conversion. The conversion of atmospheric dinitrogen, molecular nitrogen compound, especially atmospheric dinitrogen into that one atmospheric dinitrogen. In biological nitrogen fixation, the nitrogen is reduced to ammonia without involving any living cell. Okay, what is that word in the here, the biological and abiological nitrogen fixation. The conversion of molecular nitrogen into compounds of the nitrogen, especially ammonia, is called nitrogen fixation. Nitrogen fixation, nitrogen fixation is a reduced processing. What is that is called? Nitrogen fixation will stop the if there is no reducing conditions or if oxygen is present. This nitrogen fixation may take place by two different methods. One is the abiological and biological method. First, we will discuss about the abiological nitrogen fixation. What is abiological nitrogen fixation? Abiological nitrogen fixation, the nitrogen is reduced to ammonia without involving any living cells. Hello? ఎందుకు నాన్న? నేను కాలేజీలోనే ఉన్నా. డిన్నర్ చేస్తావు కదా? And abiological fixation can be two types. One is the industrial and the second one is the natural. Abiological fixation, two types. One is the industrial, second one is the natural. Industrial nitrogen fixation. First one, for example, industrial. This is the in the Haber's procedure. This nitrogen is called the Haberland procedures are there. Haber's process. Haber's process, synthetic ammonia is produced by passing a mixture of nitrogen and hydrogen. See here, you can observe. This is the industry level to fix in this one. One is the nitrogen fixing in the Haberland, nitrogen and hydrogen through a mixture of the nitrogen and hydrogen through a bed of catalysis. That is the iron oxidized at a very high temperature and pressure. This is the nitrate N2 plus 3H2 hydrogen, nitrogen and hydrogen. The temperature is 500 degrees centigrade temperature. The atmosphere pressure is the 1000 pressure to form to the ammonia. This is the industrial fixation. This one is the industrial fixation wherein nitrogen get reduced to the ammonia. Okay, this is a industrial to processing all of in the laboratory conditions. In natural process, nitrogen can be fixed, especially during electrical discharge in the atmosphere. Thunderings, you know that one? Thunderings, storms, in that one. Such discharging in the atmosphere. It may occur during lightning storm when the nitrogen in the atmosphere can combine with oxygen to form oxides of nitrogen. See this one, atmospheric nitrogen N2 plus combined with oxygen to form 2NO. This one to oxygen again to form 2NO2. This is oxidation of nitrogen may be hydrated and trickle down to earth as combined nitrate and nitrate. Here, this is a abiological process. Next one is the biological nitrogen fixation. Biological nitrogen fixation, chemically this process is same as biological, abiological, same as the abiological. Biological nitrogen fixation is reduction of molecular nitrogen to ammonia. Reduction to molecular nitrogen to ammonia by living cell in the presence of enzyme called nitrogenase enzyme. See, nitrogen fixation in the biological different there. The first one, nitrogen fixation by free living organism and some symbiotic nitrogen fixation. Nitrogen fixation is nitrogen fixation is distinct property base proposed by a select group. Select group of organism. Because of this presence of the enzyme nitrogen in them. The process of nitrogen fixation is primarily confined to microbial cells like bacteria and cyanobacteria. These microorganisms may be independent and free living bacteria. See here some free living microbes which fix nitrogen. First one is the Clostridium free living bacteria. Status anaerobic bacteria. This is the Clostridium is a one type of bacteria. This one is a anaerobic bacteria, non photosynthetic bacteria. Here photosynthetic or non-photosynthetic bacteria. Klebsiella, facultative bacteria. This one also non-photosynthetic bacteria. Third one is the Azotobacter, aerobic bacteria, non-photosynthetic bacteria. Fourth one, Rhodospirillum, purple, non-purple bacteria, purple and non-purple bacteria photosynthesis. Anabina, cyanobacteria. Anabina is the cyanobacteria. This is a photosynthetic. See here in the table, three are non-photosynthetic bacteria. Uh, uh, Two are the photosynthetic. One is the Rhodospirillum rubra. Second one Anabina. These all are the two. Three is the photosynthetic. Two is the non-photosynthetic. Three. Two are the photosynthetic bacteria. Some Sujata, ఏంటి సుజాత అది? Uh, some, mi some microbes may become associate with the other organism and fix nitrogen. The host organisms may be lower plant or higher plant. The host organisms and the huh? ఎవరో ఎవరామె ఏం గోలలో నాకు వీళ్ళకి డిస్టర్బ్ అవుతుంది. In nitrogen fixation is the here some microbes may become associated with other organisms and fix nitrogen. The host organisms may be plant or higher plant. The host organism and the nitrogen fixation microbes established a special relationship which is called the symbiosis, and this result in the symbiosis nitrogen fixation. See how many symbiosis nitrogen fixation you can observe the table. The first one is lichens. You know lichens? Lichens is a cyanobacteria and fungi. In the lichens, both are there. Bacteria is there and fungi is there. Next one, bryophyta. Bryophyta, cyanobacteria and the symbiosis, cyanobacteria and anthocyrus. Third one, pteridophyte, cyanobacteria and azolla. Fourth one, gymnosperms, gymnosperms, cyanobacteria and cycas. And in the gymnosperms, the cyanobacteria and gymnobacteria and cycas, angiosperms, legumes and rhizobium, angiosperms, non-leguminous plant and actinomycetes. Here such as ulmus, ఇది అల్నస్ కాదు అల్మస్, అల్మస్, మైరికా, పర్షియా. These are the angiospermic. Another angiospermic, Brazilian grass. This is the Digitaria. Brazilian grass is called the Digitaria, corn and azospirillum. These are the both symbiotic bacteria. How is the mechanism of biological fixation of nitrogen? Nitrogen biological fixation, the first process, the molecular nitrogen. The first one, molecular nitrogen is a strong reducing power to reduce nitrogen, like reduce is the FAD. FAD meaning flavin adenine dinucleotide. FAD is called flavin adenine dinucleotide and reduced to the NAD. NAD nicotinamide adenine dinucleotide. NAD is called nicotinamide dinucleotide is called the that is called the nucleotide. Next one, dinucleotide. A source of energy. In this one, energy also required. A source of energy, ATP, to transfer hydrogen atoms from NADH2 or FADH2 to dinitrogen. Next, fourth one, enzyme nitrogenase. Enzyme is a nitrogenase. Fifth one, compound for tapping the ammonia, for, tapping ammonia formed since it toxic to cell. Toxic to cell. This is the fifth one. Next one, coming to the overall the reducing agent NADH2 and FADH2, FA and ATP. These are all provided by the photosynthesis and respiration. You know, in the photosynthesis, the light reaction to produce the assimilatory powers. The assimilatory powers name is called NADH2, FADH2, and ATP. These are all called assimilatory flowers. In respiration time also, in the glycolysis to produce in the total respiration also to produce the assimilatory powers. The overall biochemical process involves stepwise reducing of the nitrogen to ammonia. And how to form the nitrogen to ammonia. The enzyme, nitrogenase enzyme, here two proteins are most important. One is the molybdenum and second one is the iron. Very, very important, these two protein, molybdenum and iron protein containing protein and bind with molecule of nitrogen, that is called the N2. The N2, this nitrogen, it is the binding site. This molecule of nitrogen, this molecule of nitrogen is then acted upon by hydrogen from the reduction of coenzyme and reduced in the stepwise manner. In the first produced is the diamide. N2H2 is called the diamide and hydrazine. N2H4 is a dihydrazine. Finally, to got the ammonia, that is called 2NH3. NH3 is not liberated by the nitrogen fixer. It is toxic to the cells, and therefore these fixers combine NH3 with organic acids in the cell and form of amino acids. The general equation of nitrogen fixation may be, this is the equation of nitrogen fixation. N2 is a nitrate plus 6 ATP molecule utilize 8 protons, 8 electrons to form the 2NH3, 16 ADP plus 16 phosphate. This is the processing. Here, molecular nitrogen is a very stable molecule. Therefore, sufficient amount of cell energy in the form of ATP is required for stepwise reduction of the nitrogen to ammonia. How to these are one? You can observe to the in legume plants. Legume is called the, Fabaceae family, Leguminaceae family. Okay. In the legume, nitrogen fixation occurs in specialized bodies called, this specialized body name is called root nodule. Very, very, very important. Root nodules where you can observe. The nodule due to interaction between the bacteria. That bacteria name is called rhizobium bacteria and the legume roots. Legume roots. These are the biochemical steps for nitrogen fixation are same. However, legume nodules process special proteins called leguminology, that is called the leg hemoglobin. The synthesis of leg hemoglobin is the result of the symbiosis, because neither bacteria alone nor legume plant alone process the protein. Recently, it has been shown that a number of host genes are involved to achieve this. In addition to leguminoglobin, a group of protein called nodelin, that is nodelin, are also synthesized which help in establishing symbiosis and maintaining nodals following functioning. What is the leg hemoglobin? You can observe to this one. Leg hemoglobin is produced as a result of interaction between the bacterium and legume roots. Apparently, the rhizobium gene code for part and legume root cell genes codes for the globin motiety. Both the code products together constitute the final protein leg hemoglobin. During nitrogen fixation function of leg hemoglobin is to act as oxygen scavenger, oxygen scavenger, so that the enzyme nitrogenase then convert into to ammonia under anaerobic condition. See, this is the picture. The finest simplified flow chart sheet biochemical stepwise in nitrogen fixation. See, this is the host cytoplasm. This one is the rhizobium nodule. This one is the leg hemoglobin. This is the leg hemoglobin. Leg hemoglobin to TCA cycle. TCA is called, that is called tricarboxylic cycle or Krebs cycle. ETS, electron transport chain. Third one, peridoxine. These three to produce the electrons, protons. Two electrons, two protons, two electron, two. Total 2, 2, 6, 7, 8 electrons, 8 protons. 8 electrons and 8 protons to form to the ATPs. Sometimes to keto acid also from, that is the uricides and amino acid. This ammonia, this nitrogen N2 to form dinitrate, N2H2 to form dihydrogen, H4, N2H4 and form the ammonia. This is nitrogen iron, molybdenum complex. Nitrogen iron molybdenum complex to form. This is the simplified flow chart of biochemical steps for the nitrogen fixation. Another one, this leg hemoglobin considered to lower down the partial pressure of oxygen and help in nitrogen fixation. However, this function is specific for legumes only because free-living microbes do not possess nitrogen fixing leg hemoglobin. Moreover, it is also not be found in cyanobacteria symbiosis with other plants, which is nitrogen fixed under aerobic condition. Next one, this is a biological nitrogen fixation. Next one, nitrate and ammonia assimilation by by plants. As I pointed out of the previous part, nitrogen fixation is confined to select microbes and plants, confined to select the microbes and plants, but all plants required nitrogen. All plants require nitrogen because it has a role to play in general metabolism. Therefore, Plants which do not fix nitrogen use other combined nitrogen source such as nitrate and ammonia for carrying on metabolic activity. Nitrate is observed. Nitrate is observed by most plants and reduced to ammonia with the help of two different enzymes. The first conversion of nitrate and nitrite is catalyzed by an enzyme called nitrate reductase enzyme. This enzyme has several other important constituents include FAD, that is a flavin adenine dinucleotide, cytochrome, NADH plus, FNADH molybdenum. This one NO3, NADH, nitrate reductase enzyme to form to the NO2, NAD plus and HC. The overall process of nitrate reduction takes place in the cytosol and is an energy-dependent reaction. The enzyme nitrate reductase has been studied in the many plants, and it is observed that the enzyme is continuously synthesized and degraded. The enzyme nitrate reductase is indissoluble. This means the increase in nitrate concentration in the cytosol induces more of nitrate to be synthesized. However, when excess ammonia is produced, then it has a negative effect on the synthesis of nitrate reductase. In plants, it has also been observed that light also increases nitrate reductase when nitrate is available. In the second step, the nitrates so formed is a further reduction of ammonia. This one catalyzed the enzyme nitrate reductase enzyme. Nitrate present in the cytosol is transferred into chloroplast or plastid, where it is reduced to where it is converted into the ammonia. NO2, 3 NADH plus H plus, the enzyme utilizing here nitrate reductase enzyme, that is a NH3 plus NADP, NADP plus. Here enzyme is nitrate reductase enzyme, is able to accept electrons from source such as NADH2, NADP, FADH2. Beside reduced pyridoxine has also been shown to provide electrons to nitrate reductase for reducing nitrate to ammonia. Ammonia so formed has to be utilized quickly by plants because accumulation of ammonia has a toxic effect. Some plants, including algae also, excess ammonia which can further be oxidized to nitrate and nitrate by microorganism in the soil or water. This is the total processing this one. See, most of reduction is the inorganic combination nitrogen. This ammonia now become major source of the amino acids. Total how many? 21 types of ammonia acids are there. These are the major source of the amino acids, which are the binding Which are the binding blocks of the enzyme? Which are the binding blocks of the enzyme and proteins? Amino acids have two important chemical group. That one is the amino group. Second one is the carboxylic group. See here. This one is the carboxylic group. This one is the ammonia group. This is the typical amino acid which functional group or respect alkyl group. Ammonia, so production, major source major source of a group. However, the carboxylic group to be provided by other organism molecules synthesized by the plants. There are two major reactions amino acid biosynthesis. Two major axes. The first one is the reduction of amino acid. It has been reductive amination represent the major part of the entry for amino into the metabolic stream in plants. This initiation synthesis of glutamic acid followed by the amino acid. This reaction amino combined with a keto acid. Amino combined with a keto acids. Combined with a keto acid. The most important keto acid uh, is the alpha ketoglutarate acid. Alpha ketoglutarate acid producing during the during the operation of Krebs cycle. See, this the keto the keto acids then undergo a enzymatic reduction amination to produce an ammonia. They produced an ammonia. This is the alpha ketoglutarate acid. Alpha ketoglutarate acid. Alpha ketoglutarate acid and NH3 gives rise to glutamate or dehydrogenate to form to the glutamic acid. Keto acid is the alpha ketoglutarate acid. Glutamic is the amino acid. Coming to the similar and another amino acid is called aspartic acid is produced by reduced amination of oxalic acid. Next one is the transmination reaction. This is the another very important reaction that is the amino acid biosynthesis. This reaction involves transfer of amino group from already synthesized amino acid. This is the ketoic acid. Alpha ketoglutarate acid, aspartic acid to form to the transferase enzyme glutamic acid. This is 2 plus oxaloacetic acid. And in the above reaction, aspartic acid as transformed in amino acid. That is the amino group. That one is the NH2. NH2, the alpha glutaric acid to synthesis glutamic acid and release the keto acid. The reaction is catalyzed by the enzyme transminase enzyme. A large number of amino acids are synthesized by the transmination reaction. Amino acid are organic molecule containing nitrogen. The incorporation of nitrogen amino group from ammonia into into keto acids the incorporation amino group from ammonia ketoic acid represent the major step of the synthesis nitrogen acids. Okay. This is the total nitrogenic acid. How to the stepwise I'll explain. Now we will go for the nitrogen cycle. Mm, mm. Next, we will discuss about the biosynthesis of proteins. How do proteins today class, we will complete total nitrogen metabolism, biological nitrogen, how to nitrogen, abiological nitrogen fixation and biological nitrogen fixation, how to nitrogen fixation is the form. These are things are the complete the one. Now we will discuss about to the genetic code. 1 second, I'll share my PPT. All nitrogen process is completely completed. Okay. Now we will discuss about the another topic, that is the biosynthesis of proteins. Biosynthesis of proteins. First one, proteins are molecular composed of one more polypeptide chains. One more polypeptide chains. Each polypeptide chain consists of a series of amino acid residues linked together by peptides. These two are linked by the peptide bond. Around 20 amino acid participate in protein synthesis. All the 21 amino acid. Now the total amino acid is the to identify the scientist 21. 21 amino acid are not present in a protein. All are not present in a protein. Generally, a protein molecule has 300 to 3000 amino acid. 300 to 3000. The nature and type of a protein is dependent on the number, nature, and sequence of amino acid, which is the determined by number, nature, and sequence of nitrogen in based on a DNA. This is the general defect. What is the genetic code? What is the genetic code? Genetic code meaning the mRNA is synthesized from DNA and based on the message protein is synthesized with specific amino acid linked by peptide bonds on the ribosome. The relationship between the nucleotide sequence in the mRNA, the sequence of amino acid in a protein is given by genetic code. That is called the genetic code. The nucleotide sequence that specify an amino acid is called code. That one is called code. That is, it is a triple code. Always genetic code, you can observe the triple code condition. The nucleotide being 4 and 3 constituting code. The possible code are the 64. Okay, 4 in 4 threes are. See, 4 into 4 into 4 is equal to 64 codes are there. There ends the genetic code is the degenerate. Each amino acid is coded by more than one code. More than one code as the total amino acid involved in protein synthesis is only 20. See, how many are there? Total, each and every code is the total amino acids also involvement is present. Okay, this one, each amino acid is coded by more than one code. See, each amino acid code by total 64 genetic code are there. Amino acid how many? 21. But each code more than one genetic, more than codes are. For each code, each amino acid is coded by more than one code as the total amino acid. Involved in protein synthesis is only 21. Okay. Hmm. It is a comma less. You can draw the genetic code. Don't put full stop, comma, like this. Capital letter, not like that. Okay. You put the comma less and not separate by natural divisions. Non-overlapping also. Non-overlapping. Non-overlapping and is universal of the 64 codes. AUZ is the interior codon. AUZ is the interior codon. So see, AUZ is the interior codon. Next one is the another one codon is the UAA, UAG, UGA. These three are nonsense or stop codon. Specific tRNA act as molecular adapter. Pick up corresponding amino acids with the help of amino acyl synthetase enzyme is presence of ATP. The protein synthesis machinery produced with great occurrence and translate the extract protein at the correct time of correct location in the cell. See, hundreds of amino acid linked together to make the proteins, one protein. How many hundreds of amino acid to make the one protein? But there are only how many types amino acid? 20 are there, but in nowadays, to recently, to fix the another new amino acid, that's why that is a total 21 amino acids are present. There are there 20 type of amino acid. Some we can make and some we cannot make. Some we can make, some we cannot make. But there are infinity combination. There are infinity combine of the amino acid. Can be hundreds or thousands or monomer long. The long chain is called the, that is called the polypeptide chain. That one is called the polypeptide chain. What is the protein synthesis? Before protein synthesis, we will discuss about the central dogma. Central dogma is called the from flow information from DNA to RNA, and to protein is called central dogma. అంటే DNA to RNA and protein is called DNA to form mRNA transcription, mRNA to translation to form to the transcription, translation is called central dogma. It is the proposed by the scientist is called the Crick scientist. Who proposed this central dogma? The Crick scientist to propose the this central dogma. And next one is the can be represented by the DNA, mRNA and protein. Sujata, నువ్వు వెళ్ళిపో, నాకు ఇంకా 10 నిమిషాలు ఉంది క్లాస్, మీరు వెళ్ళండి. This is a central dogma, okay? And the synthesis mRNA from DNA. Here mRNA to DNA. DNA meaning deoxyribonucleic acid is called the transcription. DNA, mRNA to DNA transcription. Sorry, DNA to mRNA. Synthesis of mRNA from DNA. DNA deoxyribosomes transcription. Translation also is the synthesis of a protein based on the information of mRNA in the form of nucleotide sequence, which are complementary to that of DNA from which is synthesized. Okay. Here in the protein synthesis is the process which a cell make protein based on the message contained with its DNA. However, DNA is only found in the nucleus. Which one is found in DNA? Only DNA found. Proteins are only made out of the nucleus. See, where in the found? In the nucellus. DNA where you can find out? In the nucleus only. Proteins to, where the proteins to made? Outside the nucleus. Outside the nucleus meaning what is that one? Cytoplasm. Houston, we have problem. Okay. Cytoplasm are there. Now, the protein synthesis, how do them different messages within the DNA molecule get the many ribosome outside the nucleus. Molecular cousin of DNA, RNA is carry these details. Ribonucleic acid, RNA is the most important in the protein synthesis. What is the RNA? A job of RNA, ribonucleic acid, is carry messenger from the DNA. What is the help of RNA? To carry the message from DNA. DNA, where DNA? In the cytoplasm, RNA is there. In that RNA, to carry the message from the DNA. DNA, where you can observe? In the nucleus to the ribosome is the cytoplasm. In the cytoplasm, there are three types of RNA. You know, three types. mRNA, tRNA, rRNA. mRNA is called messenger RNA. Carries a message from the DNA to cytoplasm. Carry the message from DNA to cytoplasm. This one is a mRNA function. tRNA function, tRNA amino acid to the mRNA to make a protein. Transport RNA amino acid to the mRNA to make the protein. rRNA make up ribosome, which make protein. Next, RNA, ribonucleic acid. RNA is almost exactly like DNA, except contains ribose sugar instead of deoxyribose sugar. This name contain uracil is the replace instead of the thymine is present. RNA is a single stranded. DNA is a double stranded, but RNA is a single stranded, not double stranded structure. So you can observe the DNA RNA strands. RNA is the only single strand. This is a cytosine, guanine, cytosine, adenosine, uracil. But in DNA, you can observe double strand. Double stranded method is there. DNA. Deoxyribonucleic acid, double stranded. In the uracil place, you can observe the thiamine is present. Okay, got it? RNA, DNA difference. RNA is a single strand, but nucleic acids, that is the purines, pyrimidine, guanine, cytosine, guanine, cytosine, uracil, adenine, and here uracil is present. But in the coming to the DNA, in the DNA, this is the which one is present? Thiamine is present. Okay, this is the structure. Okay, what is the protein synthesis? Protein synthesis, proteins, two steps are there. One is a transcription, second one is a translation. Transcription, genetic information from stranded of DNA is copied into stranded of mRNA. What is the transcription? The genetic information from a DNA, stranded of DNA, is copied into a stranded of mRNA. This is transcription. Translation, the mRNA with the help of the ribosome forms a chain of amino acid, evenly forming a protein based on the information contained from the mRNA. This is a central dogma. The flow chart, information from DNA to RNA and to protein is called central dogma theory. This is the order for even it is called central dogma of molecular biology. DNA to RNA, DNA copy may to come fix with the mRNA copy both together to form to the proteins. This is called the DNA central dogma. Next, tomorrow onwards, I will complete total the transcription, translation. Now I will stop the share. Okay.